This is the inaugural CCS lecture. This is an initiative of the school that we've instituted this year, listening to some feedback from a number of people who across the school have said, you know, we know there's just such fabulous research going on in other departments, but we never hear about it. And I know that other organisations such as WeHi have a long tradition of these institutional seminars, that they're very prestigious, and people really support them to hear the real showcase research that is done across different parts of the school. So the way we're going to structure this, each of the departments are allocated a week, and they choose how they'd like, who and how they'd like to highlight the research within their departments. And we'll have a few invited lectures from people outside as well. But I really encourage everyone within your departments, within your groups, to support this and make this the real showpiece that we hope it will be. So with that introduction, it gives me great pleasure to introduce our first department and first speakers for the year. It's the Peninsula Clinical School, and our first speaker is Professor Velandai Srikanth, who is the head of the school. He is also the director of the National Centre for Healthy Ageing, which is a $35 million federal government initiative to advance ageing research that is centred at Peninsula. It's absolutely exciting initiative that the school is front and centre of that. He is an NHMRC Practitioner Fellow. He's been the recipient of continuous fundings from the NHMRC and Howard Foundation for more than a decade. His programs of research span several aspects of ageing health, with a primary interest in risk factors and mechanisms for dementia, and particularly in the vascular space. He's also interested in other aspects of ageing health and gait and cognition. So Srikanth is going to kick off the session by talking about epidemiological approaches to brain ageing, and then we'll hear Associate Professor Chris Morgan, who unfortunately in this COVID hybrid world has to join us virtually because he's a COVID contact. And Chris is an Associate Professor in the department, who's going to be talking about diabetes as a model for brain ageing. Is it all vascular? So we're looking forward to both of your talks. So Srikanth.
Thank you, Jerry. Thank you for that kind introduction. So the introduction was about me and Chris doing a tag team presentation. Chris, unfortunately, is on the Zoom at the moment, so he's going to do a little bit in the middle, and I'm going to start and finish the presentation, so to speak. Now, this presentation is more about the kind of work that my group has been leading. This is not something which encompasses everything that the National Centre for Healthy Ageing is doing. There are several pieces of work within that include health services research, translational work, which I'm not going to speak about today. So I think what I'm going to talk about today is specifically with respect to the work that we've done and are doing in the field of brain ageing. When I say brain ageing, I'm talking about an array of things, not just dementia. The kind of work that we do is rooted primarily in analytical epidemiology. So I'm a geriatrician. I'm not a neurologist. I'm not an endocrinologist, but I dabble with a lot of analytical epidemiology by using techniques in data-rich cohorts. So my expertise and Chris's expertise is primarily in observational epidemiology in cohort studies. And we choose to use a number of designs and techniques which enable us to make causal inferences from our work. So we are adopting a lifespan approach of looking at brain aging. And by that, I mean not just restricting myself and ourselves to the older section of aging. We are going to be looking even going down to very young age and what that means in terms of brain aging. We can't do that in a single cohort. So what we'll be presenting to you is snapshots of evidence from different age groups of people. We pay a lot of attention to choosing our comparison groups, keeping in mind validity and generalizability. That's often an important thing if you're trying to make causal inferences. I love using technology, various kinds, particularly, and what we've tended to use is various types of brain imaging, ocular imaging, blood pressure measurements, etc., to try and enhance the richness of data that we have in our cohorts to then make enough inferences about pathways and mechanisms. And given that I started my early PhD days and my early postdoc days in epidemiological institutes, there's a huge emphasis that we place on analytical techniques such as mediation, effect modification, interaction, controlling for confounding, to try and tighten up the results that come out of our cohort studies. As you know, trials are the pinnacle of epidemiology, clinical trials, randomized clinical trials. Cohorts are the next level down. But if cohorts are done well, you can actually make some reasonably strong inferences. So that's just a glimpse into the type of work or the broad framework of work that we do. And then I'll go straight to the point about brain aging and what this might mean to different people. This is a great paper that's just come out in Nature just about a couple of weeks ago, and I recommend people interested in aging and dementia and brain aging to look at this. And this is a very large collaborative group, an international collaborative group. They pulled together data from various sources, from various studies, MRI data particularly, and developed models of trajectories of change in brain structure. Now, these are modelings, not true estimates. This doesn't particularly reflect what we think as normal, because the data are derived from different kinds of clinical and non-clinical cohorts. But you can see that if I can point you here, there are different phenotypes that they've modeled here. So we're looking at gray matter volume, white matter, subcortical volumes, ventricular volumes, etc. And each of these take a slightly different trajectory going from fetal life up to very old age. So cortical thickness grows rapidly in very early life and then plateaus off, peaks somewhere in the ages between 1 and 5 years, and then slowly does a decline further down. Gray matter volume, which is the pink bit, peaks somewhere again in early infant life, rises up again, peaks again here, and then plateaus off. So we start to lose neurons and white matter structures relatively early in life, not just in old age. And the issue is, why does that happen? What are the mechanisms underlying that? Is it all disease? Is it all part of normal? And that's part of the whole picture of what we're doing in our studies. It's difficult to define what normal brain aging is. And I prefer to look at it as aging brains in people without disease and aging brains in people with what we know as disease currently. We might find diseases and conditions later on which we're not aware of now, which could explain what we think is normal at this point. So why did we choose diabetes mellitus as a model for brain aging? So diabetes mellitus is, as you know, a cluster of things that happen. It's characterized in type 2 diabetes by insulin resistance. Both type 1 and type 2 diabetes have hyperglycemia as an important factor. Glucose metabolism is important for the brain, and glucose has important effects on the brain. There are several other things that happen in diabetes: adiposity, lipid dysregulation, glycation or advanced glycation, chronic inflammation, and all these have been postulated as individually as factors that are responsible for brain aging as well. And we know that diabetes is strongly associated with cardiovascular complications, renal complications. In fact, it's quite normal to hear people talk about diabetes as a vascular condition, even though it's not just a vascular condition. And so why not the brain as well? The next question that you have to ask is it reasonable for diabetes to be considered as a model for brain aging? What is the evidence behind that? There is really, really strong evidence behind this from several prospective cohort studies. And these are a bunch of these cohort studies, meta-analyzed, and it's very clear that the presence of diabetes mellitus increases the risk of a dementia by twofold further down the tracks. We're talking about incident dementia. And this has been strongly and consistently shown across several cohort studies. So there's enough evidence to pursue the fact that this is a good model to study with a number of mechanisms that are thrown in that are common across several conditions. An interesting fact here is that the risk of dementia in people with diabetes is higher in those with middle-age onset diabetes than late-age onset diabetes. And this comes from twin studies, discordant twin pair studies. Very cleverly, the people in these, the authors of the study picked a group of people with dementia and no dementia amongst their twin cohorts and looked back and looked at the exposure to diabetes or no diabetes retrospectively. And this is what they found. There was about a twofold increase in the odds of dementia, and that odds ratio increased much more in those with diabetes onset at midlife. So whatever's happening around the midlife to late adult life spans is going to be important in studying mechanisms. So what I'm going to step into now is the studies that we've done and are currently doing to try and look into why this might be the case. Why is diabetes associated with dementia? When is it important? What parts of the brain are affected? These are the groups of people who've helped me along the way. Chris Moran started doing this as part of his PhD a few years ago, and he continues to do this work. Chris Karianis has completed his PhD in this. Kate Landy, Rachel Climby, and Michelle Calisea have contributed to various parts of her work. And Richard Beer, in particular, has been instrumental in helping us with brain imaging analysis and brain imaging computation. And I'm going to talk to you about 3 cohort studies here. So the first one is looking at late midlife. And this is a twin study, the Melbourne T2D twin study, as we call it, METS. These are pairs of twins, one with diabetes and one without diabetes. And what we did was to throw a bunch of measurements at them, which included cognitive measurements, brain imaging measurements, vascular measurements, etc., to try and work out what is the true impact of diabetes when you control well for age itself and genetic influences. The second cohort is a larger cohort, the CDOT study, in which we have about 700 people, probably close to 1,000 now, with and without type 2 diabetes. Again, a range of measurements have been employed, and we'll talk to you about this in more detail later on. That cohort was aged in the early 70s on average. The last cohort that I'm going to talk to you about, or we're going to tell you about, is the ADNI cohort, which is based in the United States. This is an open access data set. It's an elderly cohort, which is free of vascular disease in itself to start with, and are phenotyped carefully for the presence of Alzheimer's disease, mild cognitive impairment, and normal cognition. They have a wealth of data available, including amyloid PETs and CSF, etc. And we use this to try and pursue the questions that arose from the previous 2 studies. The key part of ADNI is that their mean age was closer to 75, ranging from 80 to 85. It's an older cohort. So the Melbourne Twin Study, we recruited this sample through the Australian Twin Registry and out of a sample which had been previously phenotyped for the presence or absence of type 2 diabetes. So it was a small sample, 28 twin pairs, but still rich in terms of the ability to work out what the effect of diabetes in itself would be. They were middle-aged, so we wouldn't expect to find a huge amount in terms of structural abnormalities and cognitive deficits. So we had to use techniques which would try and work out how their brains were working. The questions we asked in this study were, in middle age, is type 2 diabetes associated with a reduction in any specific cognitive abilities, altered functional brain activation during cognitive activity, so using functional MRI, And did they have any features of neurodegeneration or vascular disease using brain imaging? So they were discordant for type 2 diabetes. The one twin clearly had type 2 diabetes determined by plasma glucose and other measurements. The other twin did not. The 28 pairs, 10 of them were monozygotic, so they were perfectly matched for genes in those 10 pairs. They had a range of cognitive tests applied, not just your standard paper and pencil tasks, but really sensitive computerized cognitive tests. We used a 3T MRI system to look at structural MRI. We also did a functional MRI looking at a visual memory task while they were in the MRI system. 3 years later, we repeated these measures, but we also added other measurements. So we added measurements of perfusion, which is arterial spin labeling, diffusion tensor imaging to look at the integrity of their white matter, and we did FDG PET, looking at how their glucose metabolism occurred in the brain. They also had eye measurements using ocular coherence tomography to look at retinal nerve fiber layer thickness. So we tried to use as many measurements to try and sensitively pick up any changes or differences in either structure or function. The type of analyses we used were within pair differences. So that's the strongest, that's the advantage of having a twin study. You can actually partition out effects quite clearly with it, and mixed level modeling, which takes care of within and between pair differences. And these are the results from this cohort. Sample characteristics. They were roughly aged about 60 years. They were really well matched in a variety of things, except for those things which would characterize people as having diabetes. So for example, their fasting glucose, the presence of hypertension, their waist circumference, their weight, being on blood pressure or cholesterol agents were clearly more in the twin with diabetes compared with the twin without diabetes. The duration of diabetes roughly in this sample of twins with diabetes was about 10 years. Here are the interesting results that came out of the study. So across a range of cognitive tests, and I haven't presented every cognitive test that we threw at them, there were no differences that came out. So the results are this, that there were no obvious cognitive score differences when you compared the two. So they performed, if you threw a standard cognitive test at them, you'd see a similar score. And structural MRI, when we looked at their structures of interest, total gray matter volume, white matter volume, hippocampal volume, the lesions that we see in MRI in the white matter called white matter lesions, they didn't have many infarcts because you wouldn't expect them to have at that age. They were also really rock solid the same. There were no differences. But what came out were clear differences when they actually did the memory task in the functional MRI. So the top row is all the pairs. The middle row is in the monozygotic twins. And in the bottom row are the dizygotic twin pairs. What's clear is that there are areas of reduced activation in the twin with type 2 diabetes compared with the twin without type 2 diabetes, but more strikingly, the red areas, there are several areas of increased activation in the diabetic twin compared with the non-diabetic twin. And the actual score that they got out of this memory test that they performed in the fMRI, again, the final scores were the same. So what this is telling us is that the way the brains work to achieve the same target for the twin with diabetes was different. We then analyzed, and these results are currently under review at the moment in a journal, looked at what happened in the cohort 3 years down the track when we measured them with several other measurements. 3 years down, we're now seeing differences in their attentional status. So the cognitive function differences are in the attention domain, how they're able to concentrate, what their distractibility is, etc. There are some differences coming out in the integrity of the white matter in certain areas of the brain. Not huge differences, but subtle differences coming out. Again, there were no differences in perfusion or aortic stiffness or other vascular measurements that we measured. There is the glimmer of a little subtle change in subcortical grey matter structure volume, particularly in the area called nucleus accumbens or area accumbens, which is a little bit of a hub for dopaminergic connections and serotonergic connections through several parts of the brain. There were no differences in the retinal ocular coherence tomography scans that we found. The striking thing that we did find was widespread reduction in glucose metabolism in the brain. So if you put that all together and take into account that this is a middle-aged cohort with not significant vascular disease to see, we find that type 2 diabetes is associated with probable neuronal dysfunction and compensatory increase in cognitive effort. There are subtle alterations that develop over a few years in white matter integrity and subcortical grey matter structure. Profound reduction in glucose metabolism, although we didn't measure glucose metabolism at the first instance in the first wave, it could have been present right then. And all these changes correlated with that reduced attentional ability that we found. Again, there were no differences in measures of vascular function, perfusion, structure, etc. So what this is telling us is independent of age itself, genes mostly, because dizygotic twins have not perfect control for genes, and early shared environment, assuming that all the twins had a very similar environment in their early life. It means that environmental factors, maybe adult factors, exposures, and maybe epigenetic factors may have a role to play in these differences that we see. I'm going to pass on to Chris to talk to you about the CDOT study and the ADNI study, which he really had a major role to play. Chris, are you online?
Yes, thank you. Hopefully you can hear me okay. So as Srikanth talked about, the twin study that he presented, just to remind you, were people aged roughly 60 years old. What we wanted to do in this cohort was to look at people who were approximately a decade older. So what we've done here, and this is two cohorts that Srikanth led and started back about 15 years ago, were people who were independently living in the community in southern Tasmania, were randomly selected from the electoral roll and asked if they were willing to participate in this study. The study used or took a lot of details with regards to cognitive performance, about an hour-long session of cognitive testing, brain imaging, and a lot of medical information. And what we really wanted to do was, first of all, unlike the twin study where we hadn't seen a strong cognitive signal, first of all, the first simple question was type 2 diabetes associated with poorer cognitive function? The second question we wanted to ask from this cohort, and Srikanth alluded to this a little bit, was about 10 years ago when people thought of diabetes and brain health, we thought traditionally that it was stroke driving all of this. Consistent with our theory about whether or not diabetes was linked to accelerated aging or as a model of accelerated aging, we were really interested to see whether or not neurodegeneration, so brain atrophy, was that driving any cognitive signals that we were finding rather than strokes or infarcts. The third question then that we wanted to answer was, if type 2 diabetes was associated with cognitive function impairment, if type 2 diabetes was associated with neurodegeneration, did this neurodegeneration explain the association between type 2 diabetes and the poorer cognitive function? So what I'm going to present over the next few slides is really summarized in a couple of different papers. So if people are after them, they're Diabetes Care and Diabetologia, and also got, or one of the papers got an editorial in Diabetes Care as well. So Srikanth, if you're happy to skip, I'll describe the characteristics of the Tasmanian study. So in this cohort, we had 350 people with type 2 diabetes and just a slightly larger number of people without type 2 diabetes. As you can see, they were older than the twin study, and those without type 2 diabetes were about 5 years older than those without type 2 diabetes. The proportions of women were similar between both groups. Similarly, the amount of education in both groups were comparable. Unsurprisingly, for people who are familiar with working with people with type 2 diabetes, they did have slightly higher scores on a depression scoring test. And again, consistent with the phenotype that you would expect to find with those with type 2 diabetes, they were on average more obese, so with greater body mass index, were more likely to have a diagnosis of high blood pressure, were more likely to have a diagnosis of high cholesterol. And again, unsurprisingly, you can see that the mean HbA1c in that sample is higher than those without. But for those, again, who are familiar with looking at people with type 2 diabetes, this cohort who volunteered, and it's not an unusual finding, people with diabetes volunteering to appear in cohort studies in their 70s often are slightly better with regards to diabetes control. So the HbA1c here is actually quite good. As you can see, obviously, myocardial infarction and stroke, we had a fairly small proportion in both groups, but again, slightly larger in those with type 2 diabetes. And the median duration of type 2 diabetes was 7 years. So this table shows the results of the cognitive testing that we performed. So off the full 1 hour cognitive test that we done, we used some data reduction techniques to summarize these into 4 different kinds of cognitive domains, which is a pretty common approach whenever we're looking at cognitive outcomes in these kinds of studies. And we grouped them into 4 kinds of groups: spatial planning function, speed function, a memory function, and an executive function. I'll bring your attention to the 3rd column, so that one labeled model 2. And what that model 2 is these are associations between type 2 diabetes and function in each of these 4 cognitive domains, adjusting for age, sex, level of education, which is a really important predictor of performance in each of these tests, as well as mood. So we used the geriatric depression scale score to adjust for that. And what we found was that people with type 2 diabetes had poorer performance in this spatial and planning domain. We found a bit of a signal for those with in the speed domain as well, but that did not reach statistical significance. And we didn't find a signal that was statistically significant for memory or for executive function. We then, to answer our second question, was type 2 diabetes associated with brain markers of disease, neurodegenerative markers, and vascular markers? So again, we have 3 markers of brain neurodegeneration. So total hippocampal volume, which is really the memory center of the brain, gray matter volume and white matter volume on the left-hand side. And then you can see the vascular markers that we look for. So infarcts, and in particular, we're looking at subcortical infarcts. We also looked at white matter lesions or white matter hyperintensities. On MRI brain, these often show up in older people who have a vascular burden, and we look for the presence of microbleeds. On the right-hand column, you can see these are the associations between type 2 diabetes and each of these outcomes. And the associations that we're showing here are adjusted for age, for sex, and for education. And for the top 3 markers, we also adjusted for total intracranial volume, which again is a really standard approach that just simply allows us to take into account differences in people's skull or head size. So as you can see here with the asterisks, people with type 2 diabetes had lower total hippocampal volume, lower gray matter volume, and lower white matter volume. As expected, we also find a signal whereby those with type 2 diabetes had a higher or were more likely to have stroke. We did not find a signal between type 2 diabetes and white matter lesion volume or with an increased burden of microbleeds. This is a little bit complicated, so I'll show you, or I'll walk you through this kind of carefully. So from the last, or 2 slides ago, you remember we showed that there was an association between type 2 diabetes and that cognitive performance, the spatial planning performance. Here, as Srikanth circling, the minus 0.79 shows what that strength of association is whenever we adjust for age, sex, education, and mood. Then what we've done here, so this is a statistical approach called mediation analysis. What we wanted to look for was if we added these brain measures of volume, did that change the association? The theory here being that if it reduces the association, there's the potential that it lies on a causal pathway and explains some of the association between type 2 diabetes and cognitive performance. So whenever we added in those volumetric measures of neurodegeneration, so gray matter volume and white matter volume specifically, we find that we reduced the association between type 2 diabetes and visuospatial function by about 45%, which is quite suggestive that this is some of the mechanism or the pathway through which type 2 diabetes is causing this cognitive impairment or this reduction in cognitive performance. Whenever we looked at the other measures, so white matter volume, infarcts, white matter lesion volume, and microbleeds, we really didn't see that those markers had as much of an influence as gray matter and hippocampal volume. Now, for those of you who are familiar with this kind of analysis, one of the common questions is, well, what happens if you add in stroke first? Is it just simply the order of which they put in? Whenever we presented it this way, just for ease of the narrative, if you like, but it didn't matter what order we placed these variables in. Gray matter volume and white matter volume explained about 45% of that association, and the other markers really didn't. We suggest that that's lying on the causal pathway. The next question then was, can we build more evidence to increase our confidence in that? And one of the strengths of our lab, particularly with Richard Bear, is our ability to do fairly detailed imaging analysis. So if gray matter volume seems to be important, can we look at the distribution of that gray matter volume? Are there areas in the brain of people with type 2 diabetes that are preferentially affected that might be explaining the links between type 2 diabetes and cognitive performance? So panel 1 and panel 2 shows the left hemisphere and the right hemisphere of the brain. And those areas lit up in gold through orange through to red shows those areas of gray matter that are being affected and driving that link between type 2 diabetes and poor visuospatial function. At an eyeball level, looking at this pattern, a lot of this looks awfully similar, but not identical to the patterns that you would see in people who have early Alzheimer's dementia. On the right-hand side on the table there, you can see the regions are specifically named. But again, this built our confidence that maybe this idea that type 2 diabetes is a model of accelerated aging might actually hold some water here, because this is looking very like, but not identical to, Alzheimer's dementia. The next slide shows the results of some longitudinal analyses that we did. So what I've presented up until this point has been cross-sectional. We were fortunate enough to follow these people up over roughly 5 years. The graph on the top left, labeled A, shows change or trajectories with regards to verbal fluency, and panel B shows verbal memory. The red line shows those without type 2 diabetes, and the blue line shows those with type 2 diabetes. So if we look at verbal fluency, first of all, on the top left-hand corner labeled A, you can see that those with type 2 diabetes, the red line is fairly stable and in fact has a slight trend up the way. That's what a common finding that we have in longitudinal studies of people who don't have dementia. The reason why it slightly tilts up is we think there's a learning effect. People get used to doing these kinds of tests and get slightly better over time at performing them. What we found, however, was that people with type 2 diabetes did not have that effect. In fact, what they're demonstrating is that they're beginning to decline over time. And this is a 5-year period of people in mean age of 70. These guys do not have dementia in this cohort, but we're still beginning to detect a cognitive change here. Panel B shows verbal memory. Again, you can see absolutely there's a practice effect, or what we think is a practice effect. That red line is increasing across time. The blue line stays flat. So although they're not declining in this particular area, they are still not showing that learning effect that the comparator group has. So what we've said here in our longitudinal cohort is that type 2 diabetes is associated with a rate of change in verbal fluency and verbal memory. Our second question then was, are we finding similar patterns in those with, or when looking at their brain volume measures, the brain MRI? Here, the pattern isn't quite so strong. We did find that there was an increase in ventricular volume. So as the ventricle increases, that's a sign that the brain is shrinking, if you like, and there's more space being taken up by the ventricle. It didn't quite meet statistical significance for that with a p-value of 0.06, but again, sort of fits a little bit with this picture that we think we're beginning to understand a little bit better. So although we find at baseline that there were differences in total brain volume, the rate of change didn't appear to increase between the two groups. So what we've done here is we've shown that type 2 diabetes is associated with poorer cognitive function, with greater brain atrophy and more brain infarcts. That this effect that we're seeing on the cognitive performance is being largely driven by brain atrophy, not so much on the stroke side of things. There is some effects of type 2 diabetes and cognitive decline over 5 years, but not so much for brain atrophy. And that this pattern of brain atrophy actually looks quite similar, but not identical to that of Alzheimer's disease. So the question then is, what is it about type 2 diabetes? Is it driving an Alzheimer's disease type process? So as we've been stepping through these projects, we've looked first of all at people in their late 50s. Now this cohort's looking at the late 60s. We're going to move to another cohort in their late 70s. This is the Alzheimer's Disease Neuroimaging Initiative. One of the nice things about this cohort is, to some degree, we don't own it, which means it's quite nice to look at this in a sample independent of what we've been looking at. For those who aren't familiar with ADNI, ADNI's main premise was to identify biomarkers that would predict people who have mild cognitive impairment, which is a precursor to developing Alzheimer's dementia, to see whether they could predict people with mild cognitive impairment converting to Alzheimer's dementia. This is a US-based cohort, and what they did was they identified about 200 people who were cognitively normal, 400 people who had this mild cognitive impairment, and 200 people with Alzheimer's dementia. Because they were very focused on this transition period, they took a lot of really detailed measurements, including brain imaging, blood tests, and surveys of health. But they wanted to keep that sample, if you like, as clean as possible when it comes to cardiovascular risk. So they excluded people who had brain changes consistent with stroke and things like that. Our question from this cohort was, does type 2 diabetes modify the association between each of these cognitive diagnostic groups? And given that we were wondering whether there's an Alzheimer's disease picture, is type 2 diabetes associated, first of all, with those markers of neurodegeneration, but then are they related to these markers that we know exist in Alzheimer's disease, namely that of beta amyloid and tau? Our second question, which I'll present later on, is looking at what about rates of cognitive decline and what might explain that. So again, if people are after more information on this, this was published in Neurology a few years ago, and the reference is there if you're after it. So I'll walk you through this. So first of all, we wanted to look at what was their association between type 2 diabetes and cortical thickness. So we'll look at the graph first of all. We've presented two things on this graph. So the first thing we have, if you look on the y-axis, you can see this is the mean cortical thickness. And on the x-axis, first of all, there are three cognitive groups: normal controls, of which there are about 200, 400 people with mild cognitive impairment, and 200 people with Alzheimer's disease. So the first thing, if you like, is the sanity check. As we move across these cognitive groupings, you can see that normal controls, their cortical thickness declines as they move across MCI and Alzheimer's disease. Then we split that up. And this is where you can start to see the numbers get quite small. But those in the blue boxes are those with type 2 diabetes being compared to those in the white box without type 2 diabetes. Broadly, across all of these categories, we find that type 2 diabetes, as you can see with the box plots here, is associated with lower cortical thickness. And this was independent of the cognitive diagnosis of whether they had normal cognition, mild cognitive impairment, or Alzheimer's dementia. Within each of these categories, the signal wasn't quite as strong statistically, and that's probably a power issue because there's a much smaller number as we start to cut those. So we did find that in the middle column, the mild cognitive impairment column, you can see there, those with type 2 diabetes did have lower cortical thickness than those without. Similar to our previous work, then, what we wanted to do is, what is the distribution of this cortical volume loss? So as you can see, we've got the different regions highlighted there on the brain. The yellow, red, and a little mixed one in column D, in panel D. Not exactly similar to that you would expect in Alzheimer's disease, and not exactly similar to that we reported in our Tasmanian sample. But again, makes us think that there is something about type 2 diabetes that's driving this process. Again, it looks a little bit like Alzheimer's disease, but not identical to. But the advantage of this cohort is it allows us to look at markers that are specific or are common in people who have Alzheimer's dementia. So this is similar graph as before, where we have white, those without type 2 diabetes, blue, those with type 2 diabetes. And what we've done here is we've looked at the smaller cohort of people, about 200 all up, who had brain amyloid imaging. We've divided up here into 4 panels: the frontal cortex, the anterior cingulate, the precuneus, and the parietal cortex. Even just looking at this, you can see it looks a little bit messy, and that's what we find as well. So even though we adjusted for age, sex, cerebellum, PIB uptake, and ApoE4 status, which is a genetic risk factor for developing dementia, and specifically the Alzheimer's type, we really did not find a signal that suggested that type 2 diabetes was driving brain amyloid binding. If we go to the next slide, this allows us to look at cerebral spinal fluid measures of beta amyloid and tau. So in the top left panel is A beta 42, and you can see that it's going downhill, and that's what we expect to find. So in Alzheimer's dementia, what we expect as we move across from normal control to mild cognitive impairment to Alzheimer's disease, we expect to see the brain amyloid binding increases because insoluble amyloid is being deposited on the brain. Conversely, whenever we look at cerebral spinal fluid, we expect to see that soluble amyloid is decreasing across that, and we think that's because it's being deposited in the brain. However, we did not find that type 2 diabetes was associated with a lower amyloid beta picture. The two panels on the bottom graph show total tau, bottom left, and phosphorylated tau, which is the most neurotoxic component or kind of tau. Again, for our sanity check, as we move across for both total tau and phosphorylated tau, from normal control through to MCI to Alzheimer's disease, you can see that it gradually increases across each of those cognitive categories. And what we find is across those cognitive categories, type 2 diabetes was associated with increasing both total tau and phosphorylated tau. So what that makes us think is that type 2 diabetes might be driving a tau process, but not necessarily an amyloid process. The next slide is us moving across to look at longitudinal analysis. So all up until this point, this has been cross-sectional only. This is a quite complicated diagram, and what we've done here, and this is based on some of the limitations about what data is available longitudinally in this ADNI cohort, is we wanted to know what is it about type 2 diabetes that was driving this signal, and in particular, were there certain things that either increase the risk of progression or decrease the risk of progression. This diagram is the kind of diagram you get with an analytical approach called latent growth curve modeling. So I'll walk you through some of that. On the right-hand side of the figure, you can see two circles. The top right-hand corner of the circle is change in cognitive function, and the bottom right-hand circle or oval is change in cortical thickness. So this is the longitudinal component of the ADNI data that we wanted to look at, change in cognition at the top and change in brain thickness at the bottom. Moving to the left of that are two further ovals, baseline cognitive function and baseline cortical thickness. So does your baseline cognition or your baseline brain volume predict or explain or mediate or modify your future change in brain function measured through cognition or brain structure measured through brain MRI? What we've highlighted here in the red rectangle and the green rectangle is that we find that type 2 diabetes was indirectly associated to changing cognition, so cognition over time. And what it was doing that via your baseline brain volume. So what we're getting here is a much more complicated picture, and Srikanth will talk about this later on, whereby type 2 diabetes seems to be acting on what brain volume you currently have, and they're working together on future cognitive change. However, it's not all pessimistic. Below that, at the bottom left-hand corner of that red box, you can see that we find an interaction whereby higher degrees of education appear to offset the negative effect of type 2 diabetes. And it did that by giving you better brain thickness at baseline, which protected against change in cognition. So summarizing that complex diagram, there was an indirect pathway linking type 2 diabetes with cortical decline via its baseline cortical thickness, and that education appeared to have a protective effect. And we tend to wonder whether or not this is a sign of cognitive reserve or brain reserve. So in this ADNI cohort, we find that type 2 diabetes was associated with reduced cortical thickness, increased total tau and phosphorylated tau in cerebral spinal fluid. It did not appear to be driving a CSF or a brain imaging measurement of beta amyloid. There was an indirect association with cognitive decline, and this seemed to be via baseline cognitive function, and that poor education amplified it and better education seemed to offset it. And this was independent of the diagnosis of mild cognitive impairment, Alzheimer's dementia, or normal cognition. The question then was, what are the mechanisms that are driving this increased tau production in people with type 2? I might stop talking there and hand you back to Srikanth.
Thanks, Chris. We've only got a few minutes before questions, so I'm going to rush through this bit. This is a schematic that we put forward recently to try and tie in why this might be happening. So if you look at the expression of cognitive dysfunction as the end outcome, and the preceding threats to that is neuronal dysfunction and loss, then there could be several mediators that live within the cluster of diabetes that we could consider as pathways and mechanisms, and several modifiers that we can measure in observational work that could be influential factors in that causal pathway. So really what we have is a strong suggestion of a direct effect on neurons, some suggestion of indirect effect of cerebrovascular disease, except consider that the cohorts that we've studied don't have a huge prevalence of cerebrovascular disease. So if you do find more accrual of stroke, then that clearly will lead to cognitive decline. So you can't exclude vascular pathways. It's a question of when they come into focus. You can't ignore, and this is something which might be interesting to consider in the future, that there is a reverse effect of cognitive change or structural brain change on the expression of type 2 diabetes or amplifying the type 2 diabetes state. So reverse causality is still an issue here. We've done a few bits of work trying to pursue evidence for mechanisms within the context of epidemiological work. We've showed cross-sectional associations of glycation end products with measures of brain atrophy. Chris has worked with a very large group in the US, and he's showing, still under review, that the degree of hyperglycemia has a relationship to brain structure and the risk of dementia in the future, and that might carry a bit of a U-shaped relationship. That's still a work in progress. We attempted to get a study up and going looking at neuroinflammation in the brain, comparing people with and without type 2 diabetes. We'd recruited the sample, we had the scans and the FEMPA ligands ready to go with Gary Egan's unit, but we had to put a stop to it when COVID hit 2020. So that's still on the cards and still something interesting that we might pursue. Future work, we're involved in some work looking at available data through the UK Biobank. We're looking at the question, are there profiles of cardiometabolic health that better predict risk of aging? So it's not everyone with type 2 diabetes, I suspect, or cardiometabolic influences that's going to develop accelerated brain aging. Are there groups of people within there that would be more at risk? And just looking at the effect of sex and menopause, we're finding very early that there are sex and menopausal dependent latent classes in that sample which have different levels of association with brain structure. So that's something to watch. We've just been awarded, together with collaborators in the US, Eva Feldman from the University of Michigan, who I think Mark Cooper introduced me to at a conference. We're looking at studying type 1 diabetes and hyperglycemia and variability in glycemia as a mechanism, but we're looking at a really unique cohort that the Murdoch Institute has developed over time, which is now reaching the age of 30 years from very young childhood. And we're throwing a whole lot of imaging and cognitive work in the mix to try and understand the effect of glucose and its on the brain. So I'd like to stop there because we've only got about 10 minutes to go for questions. And I'd like to acknowledge our funding sources particularly. Thank you.
Well, thank you, Srikanth and Chris. That was a absolutely fabulous way to kick off our CCS seminar series, and you've set a very high bar for the departments to follow. So, any questions? Marshall.
Great talk, Srikanth. Do you see the same changes in bad type 1 diabetes because you have a longer follow-up? And I'm just trying to understand whether this is bad glucose or cerebrovascular inflammation playing a role.
Type 1 diabetes, there's been a lot more work done within the context of the glucose control trials on cognitive function. And the story that comes out there is all these trials were directed to intensive glucose lowering, right? And that didn't have an effect on cognitive function, but there were younger cohorts, really. It did have the reverse effect where there were more adverse events coming through. So there's not good evidence at this point in time that it's all glucose related. So it goes towards answering your question, is it all glucose related? But I think there is substantial evidence coming out saying that the variability in glucose might have an impact on brain structure. There are brain structural changes in younger people with type 1 diabetes that are not dissimilar. So the white matter integrity is an issue. The effect of significant hypoglycemic events is coming out as an important issue. So clearly there are different pathways, and I'm hoping that, so in answer to your question about inflammation, it's really hard to measure neuroinflammation. I think Terry probably know more about that than I do. It's easier to measure peripheral inflammation, but the two don't equate.
Yeah.
What you measure in the periphery is not the same as what you might measure in the brain. And Helmut might have a lot more to add to that as well.
Robert.
Exercise is one of the measures that you consider, broadly speaking, so brain volume.
Yep.
Is there any difference for diabetic and non-diabetic in the amount of exercise?
There are some papers that have been published that describe the effect of physical activity on certain brain structures, particularly the hippocampus. So hippocampal volume seems to change a little bit with physical activity and exercise.
Well, what's his name?
In terms of work on animal models, there's been work been done in Queensland particularly, I think, showing that there is a strong relationship between exercise and hippocampal function. In our cohorts, we couldn't find a modifying influence of physical activity, although the measurements of physical activity were thermometer-based, which is not as sensitive as others that you can use now, or questionnaire-based.
Nicole.
So the physical activity, though, was a beautiful talk, and I
wasn't bent before.
Understood it, so that's a very good sign. So my question was about your first hypothesis.
Can't see anything on the X-ray.
You have the data within the same cohorts that you described in terms of inheritance disease.
We have indirect markers of compliance, and obviously HbA1c is a measure, but it's never good enough to really answer that reverse causality hypothesis.
There's something there, but it's not.
The truth is, if you you really have to go further back in time.
All right. I think he might have
younger age.
I think he's got a break
diabetes
at the base of his
developed to be truly able to answer the question:
proximal phalanx.
Is it just two things traveling parallel together? Is the diabetes happening because it happened and it happened in the same person who's having
I think he's got a
accelerated brain
fracture.
aging?
He does. He's got a fracture at the base of
The
his
two
proximal
actually
phalanx,
come out of each other's
which
path.
is causing deviation.
It's really
if you
hard
look on
to
the
look at that.
lateral,
From an epidemiological
you can see
viewpoint,
that there's a.
yeah, we had a very simple measure of renal
Why
function.
don't
which
I
is
get in touch
serum
with the registrar
creatinine.
and they can sort that out over the weekend?
They
Fine.
had very little in terms of
Is that all right?
obvious clinical history of
sure.
renal disease.
So
these were largely people who were well
I'll get
controlled.
who's on call.
The
and they
ADNI
can give
cohort's
that.
different. They didn't have any
needs some
data
manipulation,
on
and
renal
then
disease.
Not
But there are
anything like that. But
there are
if
data
you go back and look
suggest
at his
that the
X-ray,
presence of chronic
you got his
kidney
X-rays up.
disease is associated with cognitive aging,
Yeah, so
cognitive
you can give
dysfunction.
him that.
So it's not a bad idea to think that it might be on the causal pathway, but whether it's actually the same mechanisms involved or not is another question. So we can't look at
You've got
the
the
blood-brain barrier very easily.
So if you look at
And
the
that
AP,
probably is a site
look at
which
the AP, you can
is
see
truly
the
part which
and then
connects up
more-
vascular supply, nutrients, etc., to the neuronal health part of it. I'm not sure there are good ways to measure it in vivo, except there are MRI measures such as gadolinium measures which can be used to measure it. I can't be confident that we're missing really true microvascular disease. The measurements that we have, such as the white matter hyperintensities, etc., they reflect small vessel health, but not truly micro vessel health. So I can't be sure.
So is there any place for sort of post-mortem analysis here? Would that be?
Yep. So the Chicago group, the Rush University group, have done a series of autopsy studies. And what they found in people who die at very old age with diabetes and no diabetes, that there is really no increase in the features of Alzheimer's disease, such as amyloid, but there is a higher prevalence of infarcts and vascular disease. But these are survivors who had had autopsy. They're not the classical people that you would follow through middle age in the kinds of cohorts that we do. They are true survivors.
Toby, I think you had a question. The question there?
Yeah, sorry, I was just wondering if you look at the impact of treatments like metformin, for example.
We haven't, but there are several groups looking at that. There is a trial of metformin currently underway, being led from New South Wales. There are some indirect observational data that others have published suggesting that there may be a benefit from the use of metformin, but it's inconsistent at this point in time.
All the patients on the same treatments. For the diabetes patients.
Pretty much every person with diabetes, type 2 diabetes in our cohorts would have been on metformin at least, if not plus something else. So we wouldn't have been able to dissect out that difference in our cohorts.
Hi, there's a question from non-clinician. Well, I'm wondering whether mechanistically, could you retrieve these cerebral spinal fluid from these patients and actually measure glucose concentration to perhaps understand whether there is actually glucose enrichment in the brain of these patients with diabetes that could be brain damage and memory.
I don't think anyone's actually looked at the relationship between CSF glucose and these outcomes in such studies. I might be mistaken, but I don't think anyone has. And I'm not sure how to interpret that information myself. So it's a great idea, but I'll have to be advised by others who have more knowledge about it, CSF glucose, and how that's related to glucose metabolism.
You'd expect to be elevated anyhow in the diabetic group, so how do you know the association is the challenge. Helmut.
I think you probably alluded to the answer, but just to clarify, this increase that you see of Alzheimer's disease in diabetics, I think that's actually usually mediated by the diabetes lowered the threshold for developing symptomatic Alzheimer's rather than into the same amyloid or tau pathway.
Okay. The breadth of our data in different age groups suggests that it's an additive effect. So whatever diabetes, if it does have effect in the brain, it's adding to what's accumulating in the brain. So it's a cumulative threshold. So you've got a threshold for clinical expression, and people come to that threshold much earlier than they should. The reason why we say that is, even if we saw differences in tau between people with and without diabetes, the magnitude of the difference in tau didn't increase with the cognitive states from normal to AD. If that did increase, there was a true interaction, then you'd be thinking, is it driving a causal effect? And tau is ubiquitous, and it can be elevated in several conditions. So I think that's more a logical conclusion, much like high blood pressure, much like strokes, etc. Rather than everything being connected through an Alzheimer's pathology pathway, I think it's a cumulative effect of several conditions that reduce your threshold for expression.
All right, well, we come to the end of our time, but I'll use the chairman's authority to eat into lunch just for an extra minute and ask one last question. I know Meg Law, if he was online, would be bursting to ask this question, so I'll ask it for him. So there's a lot of interest, as you would know, in the Alzheimer's field about lymphatics, perivascular spaces as a marker of that. Have you or others looked at perivascular space load in patients with diabetes versus not diabetes?
No, we haven't compared that between people with and without diabetes, except in the first two cohorts, the prevalence of perivascular spaces was really, really low. We haven't
You looked
done
for them specifically, did you?
Yeah, we did.
Okay, because you have to look specifically.
And in the last cohort, we haven't done that.
Mm-hmm.
There are others who are looking at perivascular space burden, but I suspect that it's not going to be. If we found a strong signal with amyloid, etc., then we'd be able to draw that conclusion much more strongly. I'm not sure that it's going to give us the answer here more than what we have. worth looking at.
Yeah, absolutely. I'm sure men would be keen to collaborate. Well, thank you again. As I said, that was absolutely spectacular way to kick off our series. We're back in 2 weeks' time. We don't yet have a locked-in department to speak. So those departments that Mary and Michelle are negotiating with, please put your hand up to showcase your department, or otherwise Peninsula is going to be clearly the front runner in terms of the quality of work presented at this in this forum. So thank you, everyone. Thank you, Srikanth.
Thank you. Thanks,
Chris, and
Chris.
everyone for attendance.